Okay, hello Bio. We're continuing on with Chapter 11. We are on Section 3 now. This is going to be broken up into two parts, and this section is titled Exploring Mendelian Genetics. So, Mendelian genetics is just genetics discovered by Gregor Mendel. So, he did a lot of work, early on with those Punnett squares and everything. However, that was not enough for him. He wanted to keep going. He wanted to build on segregation. So, we're going to see what he did next. So our principle that we're going to look at today is called independent assortment. So that's going to be important to know, independent assortment. And how Mendel went about looking at independent assortment is he wanted to determine if the segregation of one pair of alleles affected the segregation of another pair of alleles. And so that's just saying that he wanted to know if tall was connected to wrinkly seeds, or if tall was connected to yellow seeds, or something like that. So in order to do that, he performed a two-factor cross. So we've been looking at one-factor crosses the last couple days, so he did a two-factor cross. So therefore, Mendel crossed true breeding plants that produced round yellow peas. So that's our genotype, big R, big R, big Y, big Y. Again, being true breeding, meaning that they're going to be homozygous. And then he used true breeding plants that produce wrinkled green peas with the genotype little r, little r, little y, little y. So that they're always going to produce wrinkled green peas, and the first one is going to always produce round yellow peas. And so all of his offspring produced round yellow peas. So remember, they had the same phenotype as our first cross, but they have different genotypes. So then after he did that, he's going to breed those two. So here is what his original two-factor cross Punnett square looked like. Again, it looks a little more complicated, however it follows all the same principles. You can see we have big R and big Y, and we will explain how to do this in class. But you can see he just takes one letter from he takes one letter from the one on the left and one letter from the one on the right and puts them together. It's just basically the exact same processes, but again, we'll practice that more in class. And so then after he performed this cross right here, he went down to make another two-factor cross by crossing these two heterozygous plants with one another to determine if alleles would segregate from each other in the F2 generation. So now our next cross is going to be big R, big Y, little y. So then we have big R, big Y, little y. And this is our Punnett square that we would expect to see, and it's going to predict a 9:3 to 3:1 ratio in our F2 generation. So you can see here that we have a lot of variation that's going to show up in this cross. And so Mendel, after looking at this Punnett square, he had some seeds that were round and yellow, some seeds that were wrinkled and green, some seeds that were round and green, and some seeds that were wrinkled and yellow. And as you can see, that's exactly what happened when he that's exactly what he predicted would happen inside his Punnett square. And so therefore, these alleles for seed shape segregated independently of those for seed color. And this principle is known as independent assortment. So there that important term is there again. And this states that genes segregate independently and they do not influence each other's inheritance. So as we can see here, the wrinkly seeds aren't connected to being green, or green seeds aren't connected to being smooth. We have a big variation here that and that's important. That's showing that none of our traits are really connected to one another. They all kind of sort independently from one another. So that's very important, knowing that they do not influence each other's inheritance. They do it on their own. And Mendel's experimental results were very close to the 9:3 to 3:1 ratio predicted by the Punnett square, and therefore he had discovered the principle of independent assortment. So Mendel, he discovered a lot of things. So he had independent assortment, he had the dominant versus recessive. So we're going to break down everything that he did. Oh, no, we'll get there in a second. So our principle of independent assortment states that genes from different traits can segregate or just separate independently during the formation of gametes, which are again our sex cells. Independent assortment helps account for the many genetic variations observed in plants, animals, and other organisms. So this does not just apply to our pea plants. It applies to animals, it applies to other plants and other organisms as well. So now we will get to a summary of everything that Mendel came up with. So our first principle that Mendel had, Our genes are passed from parents to their offspring. That's pretty basic. I think we all knew that. We're not going to get anything unless our parents gave it to us. And if two or more forms, alleles of the gene for a single trait exist, some forms of the genes may be dominant and others may be recessive. So that is just stating that one gene is pretty much going to overpower the other is basically the best way to say that. And in most sexually reproducing organisms, each adult has 2 copies of each gene, and these genes are segregated from each other when gametes are formed. And then the alleles for different genes usually segregate independently of one another. So those are a summary of his principles. I would know those are going to be pretty important. So if you have any questions, write them down and let me know.